Aging might be the secret weapon against cancer, according to a groundbreaking study that challenges everything we thought we knew. But here's the twist: older mice have fewer and less severe lung tumors than their younger peers. Researchers at Stanford University have uncovered a surprising phenomenon in the world of cancer research. In a study published in Nature, https://www.nature.com/article/S43587-025-00986-C, they found that aging mice develop significantly fewer and less aggressive lung tumors compared to their youthful counterparts. This finding contradicts the widely accepted idea that cancer risk escalates with age, mirroring a trend observed in extremely elderly humans where cancer rates seem to stabilize or even decrease. "It's quite remarkable," exclaimed Dr. Monty Winslow, Associate Professor of Genetics and Pathology. "We'd anticipate older organisms to have more frequent and severe cancers, but the study reveals otherwise." So, what is it about the molecular changes associated with aging that inhibits cancer growth? For decades, scientists have operated under the assumption that cancer risk increases with age due to the gradual accumulation of genetic mutations. In humans, cancer cases skyrocket after age 50 and peak between 70 and 80. However, beyond 85, the risk seems to stabilize or decline, leaving researchers puzzled as to whether this is due to reduced screening or protective biological mechanisms in the elderly. The Stanford study suggests the latter. Professor Dmitry Petrov, the study's senior author, explained, "The traditional cancer model suggests that aging leads to the accumulation of harmful DNA changes, eventually causing cancer. But after a certain age, aging appears to transform into a broad cancer-suppressing mechanism." To investigate the age-cancer connection, Dr. Emily Schuldiner, a former Stanford graduate student, engineered mice with fluorescently tagged lung cancers triggered by an inhaled gene delivery system. She compared young mice, 4 to 6 months old, with older ones, 20 to 21 months old, roughly equivalent to humans in their 70s and 80s. The results were astonishing. After 15 weeks of inducing cancer, the younger mice had approximately 3 times more tumors, which were also significantly larger. The younger mice had more advanced cancers in every measurable aspect, Schuldiner noted. The study also investigated 25 tumor-suppressor genes, which typically prevent cancer development. While inactivating these genes increased tumor incidence across all ages, the impact was far more pronounced in younger mice. PTEN, a standout gene, exhibited a fascinating behavior. When inactivated, it led to much more aggressive cancer growth in young mice compared to older ones. This indicates that the effect of specific mutations or targeted therapies might vary between young and old individuals, Schuldiner elaborated. Intriguingly, further analysis revealed that even cancer cells from aged mice retained detectable molecular aging signatures, despite their rapid division. However, when PTEN was inactivated, these aging signatures vanished, making the old cancer cells resemble younger ones molecularly. Dr. Winslow emphasized the importance of incorporating aging effects into cancer models for effective treatments. "Our models must reflect reality for new therapies to succeed. This study suggests that solely using young animals may neglect critical aging-related factors," he cautioned. Professor Petrov added, "The implications are potentially massive. Aging might have a beneficial aspect we can leverage for improved cancer therapies." And this is the part most people miss: could aging hold the key to unlocking more effective cancer treatments? The study opens up a new avenue of exploration, but it also raises questions. Are there other factors at play? How can we translate these findings into tangible benefits for cancer patients? Share your thoughts below, and let's continue the conversation.